There's an old Saturday Night Live skit involving Christopher Walken and Will Ferrell where they're playing in a band and Christopher Walken's demanding that there's more cowbell and that they explore the space in a musical sense. And in this lecture, we're going to look at how you can get way more ATP within the space of the mitochondrial membrane through oxidative phosphorylation. ATP synthase is a complex of subunits that's going to be responsible for making ATP in the matrix. And you'll see that there's a conformational change that's going to be important to bring ADP and inorganic phosphate together. We'll define the PO ratio for NADH and FADH2, and this is going to be how many ATPs are made per electrons that are passed from either of these molecules to the electron transport chain. We'll then walk through a calculation for how many ATPs are made from a glucose molecule. And if you learned in your biology classes that it's 36, you'll see that it's really not quite that high. Just some supplemental readings if you need them, and I've also posted some videos for you to watch again on Quercus. We saw that the pyruvate dehydrogenase complex and the citric acid cycle continued the catabolic breakdown of fuel molecules in the mitochondrial matrix, and they store some of the energy found in the molecules' bonds in the form of the reduced electron carriers NADH and FADH2. These two molecules then pass their pair of electrons to the carriers in the electron transport chain, and we saw how this resulted in a conformational change in complexes 1, 3, and 4 that set up a proton gradient in the intermembrane space. ATP synthase is then going to use this proton motive force to drive a conformational change within its complex of subunits, synthesizing ATP in the matrix. Remember, the chemiosmotic hypothesis or theory is all based around the integrity of this inner mitochondrial membrane, holding these membrane proteins in place so that we can make ATP. And the structure of ATP synthase really is key in how it makes ATP. So let's zoom in on this dark pink blob and see how it works. ATP synthase is a complex made up of multiple subunits or polypeptide chains. Some people describe it as a lollipop-type structure where this ball part is found in the matrix and is responsible for synthesizing ATP. The F1 portion is the peripheral portion of the membrane protein and makes non-covalent interactions with the F0 portion that holds it in the inner mitochondrial membrane. Protons from the intermembrane space will enter into the rotor and bind to a ring of C subunits, and there's usually anywhere from 10 to 15 subunits. As the protons bind, this results in a rotation in the ring of C subunits that's translated up into the interior of the ball portion via a gamma subunit. In the last lecture, we talked about oligomycin inhibiting ATP synthase, and it does so by blocking the translocation of protons by binding to acidic amino acid side chain residues in the C subunits. In the F1 portion are 3 alpha and 3 beta alternating subunits that are held in place by the stator. The stator stops them from rotating with the gamma subunit that runs up the middle. So as the gamma subunit rotates, this causes a conformational change in the beta subunit so that ATP can be made. Let's take a look at this rotation and conformational change. This animation will give you an idea of what ATP synthase looks like and show you the rotation that occurs in the rotor of C subunits. In gray, you're going to see the inner mitochondrial membrane with the integral peach C subunits rotating. Protons would be coming in from the bottom, and as they bind, we'll see a rotation or movement in this rotor. This blue gamma subunit will connect the rotor to the alpha and beta subunits, where the beta subunits are shown in yellow. We can only see 2 out of the 3 are shown, and in between each of the beta subunits there would be an alpha subunit. You can see the conformational changes that are occurring in both alpha and beta as this gamma subunit rotates up the middle. This hazy green stator will hold these subunits in place so that instead of them rotating along with gamma and the C ring, they can undergo the necessary conformational changes to provide an environment for catalysis. So we can see the green and the blue ADP and inorganic phosphate flying in. The beta subunits active sites will then undergo a conformational change, and a purple ATP is going to be released. You can also check out this other video that will also show you the translocation of protons from the intermembrane space. Paul Boyer came up with the mechanism for how the beta subunits catalyze their reaction. Each of the three subunits function independently, meaning they're working on their own and simultaneously. As the gamma subunit is translating the rotation up the rotor shaft due to the binding of protons and rotation in the ring of C subunits, each of the beta subunits will be separately undergoing a conformational change between three states. On the right is an illustration of the three beta subunits, and if you were to take a snapshot at any given time, each subunit will be in a different state. This is important because it means that there will be a steady stream of ATP being produced rather than a break and 3 ATPs being released at the same time. So if we start at the top, one beta subunit will be in the open state, also known as the empty or exit state where ATP leaves. One subunit will be in the loose state where ADP and PI combine, and one beta subunit will be in the tight state where ATP is made. So we can see ADP and PI binding to the loose state. Then there will be a conformational change, or the input of energy in the form of rotational energy in the gamma subunit, and then the loose state will become the purple tight state, while the tight state will become the open state and the ATP and water will be released. The tight state will then allow ATP to be formed, and the beta subunit that was in the open state is now in the loose state, and ADP and inorganic phosphate combine. So each of these three subunits will be functioning independently and changing in conformation, and it's all due to proton binding. Every time you get a proton entering in the C ring, rotation occurs and you get a conformational change between the three states. And because they are simultaneously catalyzing different reactions, first this subunit will release an ATP, and then the top one, and then the one on the left, and the cycle will continue over and over again. Let's look at this conformational change in more detail. For this video, they've just used a different abbreviation where E represents the empty or open state, and DP represents the ADP bound or loose state, while TP stands for ATP or the tight bound state. This video will start looking at the lollipop from the membrane, where we've cut away the rotor and are looking up the blue gamma stick portion running into the inside of the F1 ball portion. So you'll see in blue the gamma subunit running up the middle of the alternating alpha and beta subunits, and we're going to cut away and only look at one of the three alpha and beta subunits, alpha on the left in red and beta on yellow on the right. Every time a proton binds, there's going to be a rotation in gamma, and you'll see a conformational change in both the alpha and beta subunits. So ADP and PI come in, gamma rotates, there's a conformational change, and then ATP is formed as catalysis occurs, another rotation, and then ATP will be released. So let's watch it one more time. ATP and inorganic phosphate bind, we get a rotation conformational change, catalysis occurs, ATP gets made, and then another conformational change to the open state so it can be released. Now this is a space-filling model, so you can kind of see the interactions between these spheres, but they're all going to be amino acid side chains that are interacting between the gamma and beta subunits. The neat thing about ATP synthase is that it can work in reverse. Instead of synthesizing ATP, we can use ATP hydrolysis to drive the translocation of protons across a membrane. And some bacteria will use a similar mechanism to pump protons across a membrane at the expense of ATP. We can use fluorescence microscopy and recombinant fusion proteins to purify parts of ATP synthase and visualize this rotation. The core subunits that we would need are the alpha and beta subunits, as well as the gamma subunit. We can also use a His tag for purification, so make a fusion protein, but we could also use it to attach the lollipop head to a cover slip to be used with a microscope slide. The end of the gamma subunit can be attached to a fluorescent actin filament via streptavidin instead of having it interacting with the rotor. You could then provide ATP and water, and as ATP hydrolysis occurs, there will be a conformational change in the beta subunits, which will be translated through to the gamma subunit, rotating it, and the bound fluorescent actin filament. These are just snapshots over time where you can see this filament rotating. Or you could also track the end of the filament over time where you would see this rotating pattern. So this just shows you what this would look like, where we would see a counterclockwise rotation, which would be the opposite to what we'd be seeing for ATP synthase when it's producing ATP. But it would also depend on if you're looking up from the membrane or down from the top of the structure in the matrix. So the rotation direction doesn't really matter. What matters is that this enzyme complex is reversible depending on what the cell needs to do, either synthesizing ATP or pumping protons. Okay, so we have 3 beta subunits that are supplying this constant stream of ATP in the mitochondrial matrix. As protons are flowing through the C ring, we see a conformational change that's occurring due to that gamma subunit that's running up the middle. Every time a proton binds, you get a conformational change according to Boyer's binding change mechanism, so you need about 3 protons to pass through ATP synthase for the synthesis of 1 ATP. Remember, this does depend on the number of subunits in the C ring, which can vary, but for our purposes, it will be 3. The problem is that ATP is used elsewhere in the cell, so we need to export it out of the matrix. There is an ATP-ADP translocase that's going to export 1 ATP for the input of an ADP, which could then be used to make another ATP. You're also going to need to bring in an inorganic phosphate, and if we look at the charges on these molecules, there's actually a discrepancy of -1. The phosphates on ATP will give it a charge of -4, while ADP has -3 charge and inorganic phosphate has a -2 charge. If you add that together, we have -4 leaving the matrix while -5 is coming in. So in order to balance out the charges, there is another proton that needs to come into the matrix, so we need approximately 4 protons coming into the matrix for every ATP that's made and exported out for use. We can use this information to calculate how much ATP will be made every time NADH or FADH2 donates its pair of electrons. As NADH passes its electrons to the electron transport chain, a total of 10 protons are pumped, 4 at complex 1, 4 at complex 3, and 2 at complex 4. When FADH2 passes its electrons through complex 2, we get less protons pumped for a total of 6. We can calculate the PO ratio, where P stands for how many ATPs are made per molecular oxygen that's reduced to water at complex 4. Remember, oxygen is reduced to water using a pair of electrons and hydrogens. Since we really need 4 protons per ATP that's being made, the PO ratio for NADH will be 10 divided by 4 to give 2.5, or approximately 2.5 ATPs are going to be made per pair of electrons coming in from NADH. For FADH2, the PO ratio would be 6 divided by 4 to give 1.5. Remember, the complexes are pumping protons into a pool of protons in the intermembrane space, so we can't really keep track of which ones come from which reduced coenzyme. But we can use these ratios to calculate how much ATP or energy can come from different molecules. Now, when you're doing a calculation, if you get a half number at the very end, just round down. You can't make half an ATP molecule. Those beta subunits would just be stuck in a bound ATP state. I've also posted a journal article, and it's not required reading, but if you're interested, there is experimental evidence for coming up with these numbers. There are exceptions to these PO ratios. If you remember in glycolysis that we also make 2 NADHs, these NADHs can't be imported into the matrix where they would donate their electrons to the electron transport chain. Instead, they're going to pass their electrons to the glycerol phosphate shuttle to get the electrons onto FAD to make FADH2. So the PO ratio for the NADH made in the cytosol is equivalent to FADH2 at 1.5. The glycerol phosphate shuttle uses glycolytic intermediates where NADH reduces dihydroxyacetone phosphate, or DHAP, to glycerol 3-phosphate. There's then a second redox reaction where glycerol 3-phosphate is oxidized back to DHAP and passes the electrons to a flavoprotein 4 enzyme that uses FAD as a cofactor. This will produce FADH2, which will then pass its electrons through to coenzyme Q, and you'll get a total of 6 protons pumped through complex 3 and 4, yielding a PO ratio of 1.5 ATPs. The different isoforms of ATP synthase, so different species will have different isoforms and different structures, which will contain different numbers of C subunits, and this may result in more protons being needed per ATP, as well as different PO ratios. The other thing to keep in mind is how much water is made in oxidative phosphorylation. We've already seen how carbon dioxide is made in the pathways as a waste product, but water is also an important waste product that's made. Whenever you make an ATP by ATP synthase in those beta subunits, you're also going to make a water molecule. So if each NADH makes 2.5 ATPs, it's also going to make 2.5 waters. The other thing to keep in mind is how much water is made in oxidative phosphorylation. We've already seen how carbon dioxide is made in the pathways as a waste product, but water is also an important waste product that's made. Whenever you make an ATP by ATP synthase in those beta subunits, you're also going to make a water molecule. So if each NADH makes 2.5 ATPs, it's also going to make 2.5 waters. When the electrons are passed from NADH through to complex 4, you're also going to make a water molecule. So the water ratio would be 3.5 for NADH, where 2.5 waters are made from ATP synthesis and 1 is made in the electron transport chain. For FADH2, we have 1 less ATP being made, so 1.5 ATPs along with 1.5 waters by ATP synthase. There's then 1 additional water that's made from its electrons at complex 4. So the water ratio for FADH2 is going to be 2.5 or 2.5 waters made for every FADH2 in these pathways. This would also be how many waters are made for every NADH made in the cytosol. So we're going to walk through a calculation to figure out how much ATP is made from a single glucose, and that's completely catabolized in the presence of oxygen through all of these pathways. And you'll see that it's not the 36 that you may have heard of before. That's the utmost limit. Based on experimental evidence, we know that it's not as efficient as we thought, and we can use these PO ratios to see how much ATP is made. We can also calculate how much water is made, and keep in mind that some of the pathways may require ATP or water, so you would have to subtract off these numbers from the grand total. And if you do ever arrive at a half molecule at the end, just round down. Okay, so let's use this table to keep track of all the high energy molecules that are made and calculate how much ATP and water is made. Starting with 1 glucose molecule in glycolysis, do you remember how many ATPs, NADHs, and waters are made or required? So we would make 2 of each. Remember, 2 ATPs are required, but 4 were made in stage 2, so that would be a net of 2 ATPs. We make 2 NADHs in the cytosol, and there's going to be 2 water molecules made at the enolase step. The 2 pyruvates from glycolysis will then enter the mitochondria and will make 2 acetyl-CoAs along with 2 NADHs by the pyruvate dehydrogenase complex. Now, what about when these 2 acetyl-CoAs go into the citric acid cycle? Do you remember what molecules are made and required? Pause the lecture now and see if you can remember before moving on. So if we have 2 acetyl-CoAs coming into the cycle is going to run twice. So we are going to make 2 GTPs. We'll make 6 NADHs, since 3 are made each time the cycle runs, so 3 times 2 would be 6. 2 FADH2s would be made, and we would actually require 4 water molecules, or 2 per cycle. We can then add up all the NADHs and FADH2s that are going to go into the electron transport chain. So we'll see that there's going to be 8 NADHs made in the mitochondria, 2 from the pyruvate dehydrogenase complex and 6 from the citric acid cycle, and 4 FADH2s, 2 from the citric acid cycle and 2 from the NADHs that were made in glycolysis. Now remember, at complex 4, each pair of electrons from NADH or FADH2 is going to make a water molecule. So if we add up 8 plus 4, we'll actually get 12 waters made in the electron transport chain. Now let's use the PO ratios to calculate how many ATPs are being made. So for NADH, we would multiply 8 by 2.5 to give 20. And for the FADH2s, the PO ratio would be 1.5. So 4 times 1.5 will give you 6. This gives a total of 26 ATPs made by ATP synthase. So we're also going to make 26 waters. We can then add up all the ATPs, so 2 from glycolysis, 2 GTP equivalents, and then the 26 that were made in oxidative phosphorylation to yield a grand total of 30. And for the waters, it would be 2 from glycolysis, minus 4 in the citric acid cycle, plus 12, plus 26 to give a total of 36 waters. So our grand total for the complete oxidation of glucose would be requiring 30 ADPs and 30 PIs, plus 6 oxygens, and this number is kind of cool because it's actually half the number of NADHs and FADH2s that were made. Remember, complex 4 uses 1/2 molecular oxygen, so you only need 6 O2s. We'll produce 6 CO2s, and do make sure you know where these are being made, but remember, glucose actually has 6 carbons, so this makes sense. We've got 6 CO2s or waste products that are being made, and we're also producing 30 ATPs and 36 waters. The review questions and tutorials will walk you through a few other calculations, and in the next lecture we'll see how much ATP can be made from fat molecules. So we've seen how the structure of ATP synthase contributes to the synthesis of ATP, where a proton gradient drives a conformational change in the rotor. And this is going to be translated up to the F1 portion, resulting in a conformational change in the beta subunits for ATP synthesis in the matrix. We can then use the PO ratio to determine how many ATPs are made per reduced coenzyme and calculate how much ATP and water is made in the pathways, where we saw only 2 being made in glycolysis, 2 ATPs being made in glycolysis, but the remaining 28 were made in oxidative phosphorylation. So really the majority of your energy is coming from the mitochondria. Check out the brain break questions on Quirkus before moving on.